The next organelle which we are discussing here is mitochondria. What is mitochondria? You have studied in lower classes that mitochondria is the powerhouse of the cell. It produces and stores energy in the form of ATP. Yes, you remember because you have just now given the learnt it in class 10th. So mitochondria was first observed in flight muscles of insects. Flight muscles, you understand, the muscles which are present in the wings of the insects, by Kolliker in 1850. The term mitochondria was given by Benda. These are cylindrical bodies, generally rod shaped, filamentous or granular, with an average diameter of about 0.5 to 2 micrometer. These all facts you need to remember. Okay, so it is, if you see the structure, coming on to structure, it has two membrane. It is a double membranous structure. You can see an outer membrane here and an inner membrane here. The inner membrane is folded into finger-like projections. These finger-like projections are called as cristae. Okay, cristae. These foldings in the inner membrane. Then they have an intermembrane space in between the outer membrane and the inner membrane. The space is present which is called as intermembrane space. The material which is filled inside the main body of the mitochondria is called matrix, and matrix does have its own DNA. This is the most important thing about mitochondria, that DNA is found in mitochondria also. Where else DNA is found? Yes, DNA is found in the nucleus, plus in animal cell it is also found in mitochondria, plus in plant cell it is also found in chloroplast. So these three things you need to remember, that in plant cell mitochondria of course is present, chloroplast of course is present, and nucleus is also present. So plant cell have more amount of DNA present in them. One type of DNA is present inside the nucleus, another type is present inside the mitochondria, and the third one is chloroplast DNA. But in animal cell we don't have chloroplast, so nucleus DNA will be present in the nucleus and it will be present in the mitochondria. Clear? This is a very important factor, and this plays a very important role. This possession of DNA in the mitochondria is forensically very important, and I will teach you what is the benefit of this kind of presence of DNA. Okay, now what else? Yes, ribosomes. See, you can see the ribosomes also here. Ribosomes are also present in the mitochondria. So this, sometimes in some literature it has been considered that actually mitochondria, some scientists believe that mitochondria in itself is a prokaryotic cell which has entered into a cell just to give a symbiotic relation, just to give some help to the original cell, because the original cell did not have any energy supplies if mitochondria was absent. When mitochondria you can see it's a semi-autonomous organelle. It can sustain itself because it has its own matrix which can be compared to cytoplasm. It has its own DNA which can be compared to the prokaryotic cell. Of course it has its own ribosome. So it is a very important organelle and can be compared to a prokaryotic cell. So some scientists believe that there is a symbiotic relationship between the mitochondria and the rest of the cell, because rest of the cell is providing it with, when you have studied photosynthesis, the light and dark reaction, before light and dark reaction of photosynthesis, the glycolysis in respiration, if you have studied, glycolysis takes place in cytoplasm. So what happens? Cytoplasm is providing mitochondria with the food material and everything which is required for production of energy. Then mitochondria is producing energy and supplying that energy to the rest part of the cell. So this can be considered as a symbiotic prokaryote which is in symbiotic condition. That means benefit of both the organelles present, both the organisms are benefited in symbiosis. Similar kind of association is being thought to be there between cell and mitochondria, and this was an opinion of certain group of scientists. Okay, although there is no scientific proof of it. It has not been scientifically proven, but some scientists believe that it can be one of the cases. So this is all about mitochondria. Moving forward, the next organelle is ribosome. Ribosome you have studied also, and you know that the basic function of ribosome is protein synthesis. Important facts about ribosome, new things about ribosomes you will learn here is that ribosome is the smallest organelle in a cell, and it can be found freely floating in the cytoplasm or can be found attached to the walls of the endoplasmic reticulum, making them rough endoplasmic reticulum. Plus, just now I told you ribosome can also be found in the mitochondria and chloroplasts also. Clear? There may be around 20,000 to 30,000 ribosomes in a cell, because protein synthesis is very much required. So we need to have large quantities of ribosomes tirelessly working throughout day and night and producing proteins for us, because we are what our proteins are, and proteins are what our genes. So it is indirectly related to everything which we do is actually controlled by the proteins. Our skin, our hairs, our nails, all are proteinaceous. Our enzymes, our hormones are mostly enzymes in nature, mostly proteins in nature, which are the product of ribosomes. Okay. So here you can see there are two subunits. In the diagram I have shown you the two subunit, one larger subunit and a smaller subunit. Generally these two subunits are making up the ribosomes, but when they are not functional, these two subunits are not together. They are present separately in the cytoplasm. They come together and form this whole complete unit, having large unit and small unit attached to each other at the time of protein synthesis only. Otherwise these two subunits are separately lying somewhere in the cytoplasm. Okay. Coming on to another important thing, there are two different types of ribosomes. Prokaryotes have their own different kind of ribosome and eukaryotes have their own different kind of ribosome. So we will discuss that. Prokaryotic ribosome are called as 70S. S is the Swedberg unit. It's the Swedberg unit, in which this is by the name of one scientist, and because they are smaller in size, so it is considered to be 70 Swedberg ribosome, 70S, and where it is found in prokaryotes. This 70S ribosome is made up of two subunits, 50 and 30, 50S and 30S. See, one important point of confusion which is generally experienced by the batches earlier to you was 50 plus 30 is 80, but it is not 80, it is 70. So you don't have to use your mathematics here, because it is not like that. 70S subunit, which is a prokaryotic ribosome, will have 50S and 30S. So although 50 plus 30 is 80, but the ribosome type is 70. Coming on to the eukaryote, similarly eukaryotes are also eukaryotic 80S ribosome. Eukaryotes have 80S ribosome, and these ribosomes are also made up of two subunits, but those subunits are, the larger one is called 60S and the smaller one is called 40S. Okay, 60 plus 40 is 100, but we are not writing 100S, we are writing 80S. So this can be one point of confusion which you all need to understand.